Good evening, my dear friends. I welcome you all to the part 1 of the Science and Technology analysis on August 7, 2022. Friends, today we are going to discuss an article titled The Creatures Which Crossed Over Sea to Find India. A lot of concepts are discussed in this D. Balasubramanian article, among which the evolutionary theory supported by molecular clock is discussed. Then the continental drift theory of the Gondwana plates has been discussed. And it has also been discussed that there was a land called Lemuria. There was an island called Lemuria, and the Kumari Gandham theory of Tamil civilization. The focal point of this article is the lemurs, a nocturnal animal which is called lories in northeast India, and it can be also found at the conjoint of Kerala, Tamil Nadu, and Karnataka. The lemurs are commonly found in Madagascar. Madagascar is an offshore island away from Africa and forming part of the continent of Africa. Madagascar has always enthralled the biologists and geographists for its diverse store of different species of animals and different climatic conditions. The lemurs are very much commonly found in Madagascar. These lemurs fossils have also been found from the Buster lignite site in Gujarat. The fossils of lemurs and the bats, ancient bats, have been excavated from the Buster site in Gujarat. How the lemurs, which are commonly found in Madagascar, has also been found in India is intriguing. The distance between Africa and Madagascar is 413 kilometers, and the distance between Madagascar and India is around 3,800 kilometers. We do not find lemurs in Africa. However, the lemurs in the form of from the family of lemurs, the lories, the distant relative of lemurs, are found in India, and lemur fossils have also been found from Bastan site in Gujarat. This explains a very important phenomenon. The first theory propounded was the theory of Lemuria by Philip Sclater in 1860. The zoologist Philip Sclater stated that the island of Lemuria, there was a very large island of Lemuria between India and Madagascar, which had submerged later on. And from this Lemuria, the lemurs, the Lemuria name comes from the name of lemurs, which are found commonly in Madagascar and India. So the theory of Lemuria, which is a submerged island, submerged means it has gone under the sea, is the first theory which proposed to explain the common feature of lemurs' presence in India and Africa. As I said, in India, lemurs are called loris. These are found in northeast India and at the conjoint of Kerala, Tamil Nadu, and Karnataka. There is the theory of Tamil civilization. The Tamil civilization has come to originate from Kumarigandam, which is the submerged island Lemuria. Pavanar, a noted Tamil historian, has endorsed this belief that the Tamil civilization originated from the island between India and Madagascar, which was Kumariganda. This is the first theory. And the second theory is the Continental Drift. As we know, the Continental Drift was happening. There was a plate called Gondwana. That plate had slid, and India formed the bifurcated plate of Gondwana, which joined with Eurasian plates. And on the joining effect, Himalayas was formed. And due to this continental drift, the lemurs had crossed over the sea to India. They did not venture into Africa because one part of the Gondwana plate was joined with South America to form another plate, and there is a bifurcated plate out of which one plate joined Eurasia, as I said, Eurasian plate. So this is the continental drift theory, the Gondwana plate continental drift theory. Hence, it can be seen that these two theories are trying to explain the commonalities of lemurs found in Madagascar and India. There is a dinosaur which had lived some 80 million years ago. All this Gondwana plate tectonics was happening around 150 million years ago. And by 88 million years ago, India had joined with the Eurasian plate. The bifurcated Gondwana plate of India had joined with Eurasian plate. The fossils of dinosaurs have been found from Madagascar and India alike. That is Laplatosaurus madagascaranus. Laplatosaurus madagascaranus, a vegetarian eating, very huge neck-wala dinosaur fossils have been found from India and Madagascar. This commonality also explains that continental drift theory of Gondwana is correct. Laplatosaurus madagascarensis, Laplatosaurus madagascarensis was a very huge dinosaur which once lived some 80 million years ago. And the commonality of the finding explains that India had indeed drifted, continentally drifted from the Gondwana plate. Then, explaining the molecular clock theory. This is the third theory. In the molecular clock theory, what is explained is that what is a molecular clock? A molecular clock explains the time of mutations from a common ancestor. There will be a common ancestor for all the animals, and from the common ancestor, the gene mutations are happening and the animals are developing different characteristics with the gene mutations. Gene mutations means changes in the gene. when the changes in the gene, that is mutation, is happening, the characteristics of the animals also change. And it takes billion years to have a change happening. For example, in the blood, there is a hemoglobin which changes every billion years by 0.52 per sample. So, the molecular clock theory tries to find how much time this evolution took. That is, the point of time of change is always constant. Maybe million years, maybe lakhs of years. However, the change happens randomly, although the point of change that is a constant. The change is constant. So, a time period of a million years or billion years is looked into to find how much time the evolution took. This is the molecular clock theory. There is a freshwater fish that is known as Pallati in Kerala, which is also found in Sri Lanka. This is the greatest example of molecular clock theory. This fish is also known as, in scientific name, it is known as Pseudonema mollusk. This fish, Pallati, is commonly found in India as well as in Sri Lanka. Then there is the case of the freshwater crabs, the Gicacinidae. The freshwater crabs are known as Gicacinidae. It can be found commonly in Southeast Asia and India. These fish, the Pseudonema mollusca and the Gicacinidae, all these have come from one common ancestor which lived in India as well as Sri Lanka. The molecular clock theory finds that from the common ancestor, a particular period of time this evolution has taken place. Although this freshwater fish is very common in India and Sri Lanka, the Pallati, this fish has a common ancestor. Similarly, the crab, freshwater crab, Gicacinidae, is having a common ancestor. It is difficult to cross over the sea to go to Sri Lanka. However, this plate theory, the Gondwana plate theory, explains that this has happened because when the plates drifted, the common ancestors were diversely thrown out into different lands and countries. Friends, I have explained the Lemuria theory, the Continental Drift theory, and the molecular clock theory to understand how lemurs are commonly found in Madagascar and India. Madagascar and India is 3,800 kilometers away. However, the lemurs are not found in Africa, which is just 413 kilometers away from Madagascar. So these are the theories which explain why this far the lemurs are found and not the nearer Africa where it ought to have been found. Friends, I will come with the part 2 of the science and technology analysis in a short while from now. Till then, take care. Bye. Have a nice evening. Friends, I already discussed about the lemurs in the first part of the science and technology analysis on 7th August 2022. Now, under the part 2 of the analysis of the science and technology column of the Hindu on 7th August 2022, I am going to discuss about the Bangalore's rock agamas. The rock agamas are large lizards having orange and black color. The rock agamas are known as peninsular rock agamas or South Indian rock agamas. These rock agamas are abundantly found in urban centers such as Bangalore. Bangalore city is fast changing and most of the habitats of these rock agamas are being converted into buildings. Even after 20 kilometers from the Bangalore post office, the urbanization creeps into the habitats of rock agamas. These rock agamas are ectotherms. Ectotherms means the cold-blooded animals like reptiles and snakes. These animals cannot live. These are ectotherms and they cannot live without the presence of heat. So they need rocky surfaces and warm, sunny surfaces where they can draw their body temperature from. They cannot have their body temperature regulated because they are cold blooded animals. The reptiles, snakes, and rock agamas and lizards are very cold blooded animals. We have geckos in our homes. The geckos are small lizards on the walls of our homes. These are known as geckos. The rock agamas depend upon the rocky surfaces. The ecological conservation needs that small flora and fauna are preserved. Flora means flowers and fauna means animals. So the flora and fauna of the ecosystem should be preserved. These rock agamas also throw light on the information on which parts of the city are warming. So these are kind of, the clocks, the climate clocks, which give an information on the warmer parts of the city, which parts of the city are warming more. Because these rock agamas depend upon the warm climate. So the scientist from the Indian Institute of Science, Bangalore, has taken a study of the rock agamas and how far urbanization has affected the ecosystem, the habitat of these rock agamas. These rock agamas live on insects, and the rock agamas are eaten by reptiles like snakes, and the raptors also eat the rock agamas. Hence, where the insects are present, the rock agamas can thrive. Therefore, the rocky surfaces, the in presence of insects, the warm climate, all are necessary for this rock agamas to thrive. This south peninsular rock agama is fast disappearing in Bangalore because most of the Bangalore outskirts are also being converted into buildings. 20 kilometers away from us, I said 20 kilometers away from Bangalore, the fast urbanization is just converting the shrubs, the herbs, and the plant surfaces into buildings. So, this is a threat to the South Peninsular rock agamas life and the habitat. They cannot thrive in this manner. While planting trees, we must also ensure that rocky surfaces are not destroyed. Friends, hope the discussion on rock agamas was very much brought home to you. It was interesting. Try to search in Google which kind of animals are these rocagamas are, or these ectotherms are. You will find many interesting more facts on this. I will come out friends with monkeypox around the corner, and around 15,000 people have been affected by the monkeypox virus all around the globe from 84 countries. And considering its seriousness, the World Health Organization declaring it as public health emergency of international concern. Now, the title of the Science and Technology column, a very nice article, Why Genome Surveillance is Imperative. Friends, with the help of genomic surveillance of the genomes of the virus, the monkeypox virus, which is having a 2 lakhs base pairs in the genome, in the chromosome of the DNA of the virus, which is 6 times more than the SARS-CoV-2 virus, which is the COVID virus. SARS-CoV-2 virus was a RNA virus, whereas the monkeypox virus is the DNA virus belonging to the monkeypox virus belongs to the orthopox genus and under the Poxviridae family. As I said, it is having 2 lakhs base pairs. Base pairs means you have already seen the structure of DNA. It will be in the structure of base pairs. The genomes are the genes containing the genetic information of an organism. These genomes are located in the nucleus, in the chromosome of the nucleus. Hence, monkeypox virus has 2 lakhs strands, that is 2 lakhs base pairs. Hence, the monkeypox virus, the genomic mutation. Mutation means some errors happening in the genomes of the virus, and the change in the gene is happening, and the evolution of the virus happens. Every organism is having mutation over millions and billions of years, and from a common ancestor, several animals have been formed. So, mutation is going on happening. And this genomic mutation is also occurring in this monkeypox virus. Earlier, the smallpox viruses had only 2 mutations per annum. However, it was believed that monkeypox virus, because it was similar to the smallpox virus, it would be having very less mutations. However, with the passing of time, we can see that around 50 mutations per year is being reported of the virus. Around 50 mutations per year is being reported of the virus. Mutations means change in the genetic structure. So, this change in the genetic structure of the virus is happening, and 50 such changes are happening. So, what is the purpose of genomic surveillance? Genomic surveillance is studying the genetic strand of the virus, that is genetic sequencing of the virus. Genetic sequencing is nothing but investigating the mutation of the virus. That is, one strand is, for example, A1 is different from the next mutation A11. A11 is different from the next mutation A2 and then B2. Therefore, the mutations are happening and the virus is evolving. Genomic surveillance is trying to find out the spread of the virus, the epidemiology with related to the virus. What is epidemiology, my dear friends? Epidemiology is the study of the spread of the disease and control of the disease. And this genomic surveillance also helps to, genomic surveillance is nothing but investigation of the mutations of the virus. It helps to make decision making regarding the outbreak. Since 1970s, monkeypox virus has been around. There are two types of viruses found, that is Congo basin virus. It primarily originated in Africa in monkeys. Therefore, the name the monkeypox virus. Congo basin virus and the West Africa virus. The Congo basin virus is now named Clade 1 and Clade 2. The West Africa virus is named Clade 2 and 3. The Congo basin virus is named as Clade 1. The West Africa virus is named as Clade 2 and Clade 3. Because you cannot assign the names of the countries to the origin, to the variants. Therefore, Clade 1 and Clade 2, Clade 3 are given. However, recently we have found that A2 strain is also occurring. A2 strain has been found because of the advancement in the genomic sequencing and the genomic surveillance. A2 variant has been found from India and US and Thailand. In Europe, the super spread means spreading like anything. The super spread is in Europe is attributed to A1 variant of the virus. So there are two major variants of the virus which is shaking the 2022 out of fear of the monkeypox virus. That is A1 variant, B1 variant and A2 variant. B2 variant is 95% of the outbreaks occurring from the B2 variant, and it is spreading throughout Europe. A1 variant is found in India, US and the Thailand, and the outbreaks related to this is related to A2 virus, A2 variant. There are 6 genomes of this virus, that is 6 mutations of this virus. 2 are found from Kerala. So this A2 variant, there are 6 genomes, out of which 2 have been found from Kerala. During the COVID-19 situation, the genomic sequencing and the genomic surveillance has reached its apex, and the genomic surveillance infrastructure is everywhere now. So it is very easy to find the genomic sequencing of the virus because the infrastructure is there now. Hence, we have been able to uncover and detect A2 virus, A2 strain which is found in India has been hitherto hidden since 2021. Now we have found out the A2 strain. With the advancement in genomic surveillance and genomic sequencing, we are able to find the strain very clearly, and now it is imperative upon us to provide a very fruitful solution to the spread of the virus. In our body, the human genome is producing a protein, a polyprotein B editing complex, that is ACOBEX 3, a polyprotein B editing complex, that is ACOBEX 3, which ties to the virus and makes it ineffective. The DNA strains of the virus have been caught by the apolyprotein B editing complex 3, which binds to the virus and makes it ineffective. And the continuous evolution of the virus suffers from any effective the dangerous effects of the virus because the human genome already produces ACOBEX 3, that is A polyprotein B editing complex. This A polyprotein B editing complex makes changes to the genome of the DNA of the virus, and its further evolution is having no major dangerous effects. Friends, so with the help of all these discussions, you can understand that how this topic of genomic surveillance is very imperative. We have just finished containing the COVID 19 virus, for which this genomic surveillance and the related infrastructure was useful. Now, for the monkeypox virus, also, this becomes useful. Friends, I have come to the conclusion of this topic, genomic surveillance. Hope it has been very much useful and enjoyable to you. Google for more information on these aspects. Thank you.